In this video, we're going to look at how you actually sequence DNA strands now. And traditionally, the method that was used is called the chain Terminator method, developed by Fred Sanger. And this method is actually still used all around the world today. And the process is fairly simple. One, you get a single polynucleotide strand. For example, you could get it through using restriction endonucleases that we talked about in the previous video, to cut up the genome like that. Two, you take your polynucleotide strand and you denature it. You break hydrogen bonds by heat. Three, to your single-stranded DNA, you add nucleotide triphosphate. So DNTPS means DNA nucleotide triphosphates. And Terminator DDNTPS. So there's two Ds because we have two oxygens being removed. And I'll talk about that in a second. Then you add DNA polymerase 1 and a primer. So first off, what is polymerase 1? Polymerase 1 is the polymerase in E. coli. Polymerases will add new nucleotides to a growing strand. So for DNA polymerase 1 to work, it needs a primer. That's it. That is, it needs DNA already there on the strand to be added to, because polymerase 1 will add to the 3' hydroxyl. If you remember from our previous videos, we said that DNA goes 5' to 3'. So polymerase 1 needs a 3' hydroxyl. Then it will add new nucleotides by removing two phosphates. These two phosphates provide the energy to make a new phosphodiester bond, and it will add on new bases based on the template strand. So here we have a G on one strand, so a C is going to be added. Here we have an A and a T. Now, what's really clever about this technique is that in addition to normal nucleotides, we have a very small amount of these DD trinucleotides, which, as you see, don't have a 3' hydroxyl. So if the polymerase adds these DD nucleotides, the process of adding new nucleotides is completely stopped. Polymerase cannot add any more nucleotides. Now, each one of these DD nucleotides has a fluorescence tag. So by having thousands and millions of copies of the same DNA that you want to sequence, all you need to do is you have to say, okay, how long is my DNA and what is the fluorescence tag on the end of my DNA? So here we have 3 nucleotides, and the last nucleotide is a C, which was a DD nucleotide that's colored blue. Then you have another blue, green, red, blue, each time going up by 1 nucleotide, so you can start to sequence your DNA. And in practice, this method is fully automated. You give your DNA to like a company that will do this, or a lot of universities have these facilities on site, and you will get back something that looks like this, where each peak is saying what nucleotide is there. You can see there's some noise down here where it's saying that there's some uncertainty, but these peaks are so high that you're pretty confident that this is the DNA sequence. So this is your output from this sequencing method, and this type of method can read up to about 1,000 nucleotides from one reaction, and we call these reads. Every time you do a reaction to do DNA sequencing, you call that a read. And to do long genomes, you basically combine different reads. So let's say you have something that's 10,000 base pairs long and you want to sequence it. So maybe your first read will be 0 to 1,000. Your second read will be 990 to 1,990. So you have that 10 base pair overlap, and you can start stitching together your genome. And so One way you could do this for an entire genome is just randomly breaking the genome with sound, which is also called sonication, doing this, and then trying to piece back together the fragments. This would take a very long time, cost a lot of money, but it is doable. However, we have moved on to what's called next generation sequencing techniques, and so we're going to look at two of them in this slide and the next. The first one we're going to look at is called Pyro Sequencing or 454 sequencing. It's two words, means the same thing. What you do for this is that you take your DNA fragment and you attach it to a plastic bead, and you do it in such a way to make sure that there's only one DNA molecule per bead. You then take the DNA that's on the bead and replicate it. So imagine that this bead is just covered with the same DNA sequence over and over and over again. Then to these beads, you add a primer, DNA poly 1, and one type of a trinucleotide. So for example, here we are adding only T dinucleotides. If the nucleotides you add match to your sequence, they will base pair and DNA polymerase 1 will add them. So you let this reaction run, then you wash away all the unbound nucleotides. Then, because the way DNA polymerase 1 works, this actually, when you add new nucleotides, you release two phosphates. And these two phosphates can interact with an enzyme called luciferase. Luciferase is the enzyme in fireflies that makes them spark. So you get a little light signal when you add DNA. And so you do this and sequentially. You add all T, see if you get a spark. Add all A, see if you get a spark. Add all G's, see if you get a spark. Add all C's, get a spark. And then go back to T. By doing this, you can roughly read 1 million different DNA strands at once, which is roughly 1,000 times faster than the chain Terminator method we just looked at. A different type of sequencing is called Illumina sequencing. Here, we attach our DNA fragments to a glass plate. Then it's kind of like the same idea where we multiply our sequence millions of times on this plate. Then we add a primer, DNA polymerase 1, and this time we add all 4 nucleotides. So in the previous, we were just adding one type of nucleotide at a time. Illumina, we add all 4. The catch with Illumina is that only one nucleotide can attach at a time. So one nucleotide attaches, and then you can shoot it with a laser, get a fluorescence signal, and see what nucleotide it was, because all 4 nucleotides have different lights. And you do this so on and so on and so on. Illumina can give you accurate reads of 30 to 300 nucleotides, so not that long. But in one run, you can get 3 billion reads, so you could do a whole genome in that time span. And with these next generation sequencing, sequencing a whole genome is actually super easy. The hard part of it is to actually patch it back together to get a complete genome. But the sequencing itself is super easy. And every time a scientist sequences a genome, it's usually placed online. And the online database of choice is called GenBank. And we are sequencing genomes at such a rate that GenBank roughly doubles every 18 months. And here are just some genomes that we have sequenced from humans, mice, all the way down to parasites and pathogens. Right? And with all this information, a new field of science has really opened up called metagenomic sequencing, where you take multiple organisms and you just compare their genomes to see what you can look at. So one way this has been used is looking at our gut bacteria, which is called a microbiome, because we have an entire ecosystem in our guts. And from this microbiome, we can just compare all the different organisms and see if we can get any trends. And just one trivial piece of information that was found out is that people who weigh differently have different microbiomes. They have different bacteria in there. And as of last year, just to show you how advanced we've become, at one institute, there has been already 100,000 human genomes sequenced. So, sequencing the genome is pretty easy at this point. Just to give you a scope of how difficult it was, the first human genome took about 13 years to sequence and $300 million. You can do it now for less than $1,000, and I think roughly 4 days you can have your results back. From this human genome, though, we have learned various things, but we're still learning. That is a lot of information to piece through, a whole genome of a human. So we are still looking for trends. But the trends we have found is that half of the human genome just consists of sequences that repeat themselves. It's just the same sequence over and over and over and over and over again. About 80% of our genome is transcribed to RNA. Only roughly 1.2% of the genome encodes proteins, which is quite shocking. Scientists had it pegged like to 30 to 40% because proteins are really the macromolecules that do all the work in the cell. So it is a little surprising that only 1.2 to roughly 1.5%, it varies, but it's not more than 2 of our genome encodes proteins. And if you look at the genomes that encode proteins and see what they are, most of these proteins are enzymes, but a third of our protein coding genes, we have no idea what they do. So it is still a big learning process. Overall, we have about 21,000 open reading frames or genes that we can make. And of all our genes, most of them can be found in any living organism. Only a very small percentage of genes are unique to vertebrates, that is, organisms with a spine. And if you compare different humans, you are different from any random human by every 1 in 1,000 nucleotide, which means that you are 99.9% identical to anyone else in the world. But we know humans are quite complex, so how can that be if our proteins are no different than like yeast? And really what it comes down to is that although we only have 21,000 genes, they can be spliced or cut in different ways to lead to a lot of the complexity we see in life. So one gene sequence can make various number of proteins. Sequencing has also allowed us to get a much better understanding of evolution. As we talked about the central dogma, if you change DNA, you change proteins. And DNA can be mutated by various means. So mismatched base pairing, chemicals through like smoking or radiation through like X-ray, or it can just be a natural error in your own machinery. But if one nucleotide changes, we call that a point mutation. And although physical characteristics, that is, phenotypes, can be quite different, for example, in these two different types of corn or in chimpanzees and humans, if you look at the genome, we're not that different. There's exactly 1% difference between a human and a chimpanzee. Again, that is surprising based on the outward appearance, but there's only 1% difference. Maize and its precursor teastonite only differ by just a few genes, a handful of genes, yet you can see they're widely different in their phenotypes. And we can also learn about human history. If we look at the genomes of modern-day Europeans and Asians, we see they have roughly 1 to 4% of Neanderthal DNA. If you want to relate that to more understandable terms, that's like if you had a great-great-great-grandparent that was a Neanderthal. That's the amount of DNA Europeans and Asians have in them. But Africans do not. They don't have any Neanderthal DNA in them, which says, European and Asian descents migrated out of Africa and mated with Neanderthals. They crossbred in some capacity. In looking at genomes, we can also look at sequence variation and see how that relates to human disease. So overall, we have about 5,000 diseases that are based on mutations, and here are just a very few of those. But of these diseases, it's very rare that a disease is based on the mutation of a single gene, that is a monogenetic disease. Most genes are very complex, so it's a mutation of multiple genes, so they're hard to understand. But with this sequencing, we are much better at screening newborns for any genetic disorders they can have, and so we can um, help to treat those diseases from the moment they're born. And also, adults who are looking to have children, we can test if they're carriers for a disease so they can know the risks before having a child. And again, going back to humans in general, we only differ 1 out of every 1,000 nucleotides. And these differences are called SNPs, or single nucleotide polymorphism. So SNP is, you pronounce that SNPs. That's where your genome differs by one nucleotide. So it can be an insertion, a deletion, or just a change. And right now, we have about 10 plus million SNPs mapped out in the human genome. And the reason why these are interesting is that we are beginning to associate these SNPs with the likelihood of disease. For example, breast cancer. If you have a SNP in a gene called BRCA2, you are more likely to have breast cancer. But this is a very hard thing to do because each SNP may only increase your rate of disease or cancer by 1 to 2%. So not all that much. So it's saying that you are more likely to get it, but not that much more likely. And so you might have heard of or seen ads for personal genomic medicine. That is, they take your genome and then they design medicine specifically for you based on your DNA. But there's so much unknown about genome and SNPs that we are very far away from that to being feasible. And that's it for this video, and I will see you in the next video.